Okay, so hi everybody. My name is Anoushka, and my specific research topic slash paper was on genetic risks for Alzheimer's disease, and I could not have done any of this without the help of my mentor Krista. So thank you to her. So just to start off, I want to give a little bit of a background information on the risk for Alzheimer's. So Alzheimer's disease is a neurological disorder affecting memory and behavior, and the disease occurs in a lot of stages. So we first have healthy, then mild cognitive impairment, and then lastly, there's also full dementia, where the Alzheimer's disease really kind of affects the person's daily life and what they have to do kind of every day. And so we can see these specific stages through plaque accumulation in the brain. And there's a myriad of factors that actually correlate to getting Alzheimer's. There's genetic and lifestyle. So regarding genetics, there's a specific allele called the ApoE4 risk allele. And this allele increases plaque accumulation, specifically amyloid plaques in the brain. Which eventually contributes to the earlier onset of Alzheimer's in people with this allele. So this just means that people with the allele have a higher chance of getting the disease earlier in their life, because most people get it sometimes way later at like 60 or 70, but these people with the allele might get it maybe at 40. And then other than genetic, there's also lifestyle factors. Basic ones such as like exercise and diet obviously play a huge role, but specifically within the data I worked with, me and my mentor, we looked at education because with people with more education, there's this thing called cognitive reserve where their brain has formed a lot of neural networks. So those kind of hold almost as a reserve when Alzheimer's does kind of deteriorate those other neural networks. And so this can help delay the onset of the disease, but again, it doesn't prevent it fully. And the way we can look at all these brain pathology indicators in the brain, we can use MRI scans and PET imaging to detect the severity of the symptoms in an individual's brain. So one of two specific biomarkers for Alzheimer's disease, which I also refer to as AD, is white matter hyperintensities, and these are a sign of cerebrovascular risk and risk for Alzheimer's. So these are specific areas under an MRI scan that are seen as a specific area where there's a lot of white matter hyperintensity, and these indicate specific changes or damages in the brain's white matter, and these can also be like dead brain tissue. So as you can see in the image right here, this image really shows very clearly how compared to mild and severe cognitive decline in an individual, the amount of white matter hyperintensities are, increasing slowly as the person gets worse and worse regarding cognitive decline. So you can see in the very severe image, there's a lot of areas of white matter hyperintensities. And then another way we can see Alzheimer's disease in the brain is through hippocampal atrophy, which also just means the reduction of hippocampal volume. So also in this image, we can see a healthy patient versus an Alzheimer's patient. So specifically, this atrophy will occur everywhere, but especially in the hippocampus. And the hippocampus is obviously where the brain does a lot of its memory making ability and problem solving abilities. So that's why that's one of the first things an Alzheimer patient may struggle with or have a lack of. And so as you can see in the Alzheimer patient visual, they have a severe reduction in their hippocampus, and there's that big black space where the healthy one has more hippocampal volume. And then talking about just the methodology that I used to do this study, me and my mentor, we used imaging markers that came from pre-processing performed by the Alzheimer's Disease Neuroimaging Initiative, otherwise known as AD&I. So as you can see on the image, this AD&I database was really helpful because it gave us some MRI data, PET data, as well as genetic data, so we could see whether certain patients had the ApoE4 risk allele or not. And then in order to kind of visualize all this data, we used RStudio, which is a IDE for the language R, to perform all data sorting and visualizations. And then within RStudio, we use the ggplot2 library to make our figures, which I'll show in the next couple of slides. So the first figure that we kind of looked at was looking at hippocampal volume in 4 different groups of cognitive status. So as you can see in the visual, there's a legend, and in that legend, we have those 4 different groups. So one group is kind of healthy to mild cognitive impairment to full-on Alzheimer's. The second group was the patient always stayed having Alzheimer's. Third group means the patient went from healthy to full-on Alzheimer's, and then last but not least, mild cognitive impairment to Alzheimer's over the amount of visits that they conducted in like their hospital. So as you can see in the light blue and the kind of purple color, like the magenta color, those two specific lines show a really downwards negative steep slope, meaning that as a patient progressed closer to getting full-on Alzheimer's or had full-on Alzheimer's, we can see that they had a severe reduction in hippocampal volume and they had a more steeper, like negative slope. So that's how we kind of associated a reduction in hippocampal volume with having the progression of Alzheimer's or having full-on Alzheimer's. And then we also wanted to look at the allele and how that played a role in these four different cognitive status groups. So here we made a graph that showed the cognitive status over time in individuals with or without the ApoE4 allele presence. So as you can see in the first line, those are people with the allele, and the second line is people without the allele. So once again, people with that purple and blue kind of exhibited showing are people with the ApoE4 allele in the yes column. They had more blue and kind of purple dots, which means that they actually had an earlier onset of Alzheimer's disease at around, the age of 50, 60, while the people without the disease kind of the purple and blue dots are more scattered later, meaning at like 70 to 80 to 90. So this kind of showed that the ApoE4 status does in fact correlate to getting Alzheimer's at an earlier age. And then just to elaborate on what I talked about earlier when I showed the MRI scans, we just wanted to prove that white matter lesions or white matter hyperintensities are increased when you have that ApoE4 allele. So as you can see here, we compared people with the allele and people without the allele. And we can see that though the medians were quite the same, kind of that line in the box and whisker plot is relatively the same, the spread is different in each of the two groups. So we can see that in the group that has the allele, the spread is kind of higher, meaning that if you did have the ApoE4 allele, you have a higher amount of white matter hyperintensity or white matter lesion volume than the group that did not have the allele. And then we made the same box and whisker plot with hippocampal volume, another biomarker that I talked about in the beginning. So hippocampal volume would actually be decreased if you exhibited Alzheimer's like symptoms. So here we compared again those two different groups, and we can see here the median slightly shifted. People with the allele had a slightly lower overall hippocampal volume than the group without the allele. And we can see also that the spread, people without the allele had way higher hippocampal volume. It reached almost the top line where it's 0.0050. And that means they had a way higher hippocampal volume on average as well. And so this shows a direct correlation between hippocampal volume and, the reduction of hippocampal volume and having the E4 allele. And then just as you saw earlier, we looked at the ApoE4 allele, but then we also looked at amyloid levels. So amyloid levels are what cause the buildup of those amyloid plaques that result in having Alzheimer's disease. So we wanted to look at how amyloid levels and ApoE4 having the ApoE4 allele compare in people without Alzheimer's. So this is looking at non-Alzheimer patients. So this box and whisker plot shows white matter hyperintensity volume across four different groups. So that first part of the label, the no/yes, that refers to whether the patient has the ApoE4 allele. And then that second part, the negative/positive, refers to the patient's amyloid status. After a certain threshold, a patient can be considered as like amyloid positive. And so here we kind of found out that the two groups on the like last two groups that both say positive, meaning that they have a positive amyloid status, we can see that they have on average a higher median than the other two groups with a negative amyloid status. We can see that groups with a positive amyloid status have a higher white matter hyperintensity volume in their brain. And that also shows that this is another way that we can kind of track people on the kind of progression of getting Alzheimer's. And then, like I mentioned earlier, it's not just genetics that play a role, of course, it's also lifestyle factors. And specifically in the AD9 data set, we were able to look at education and see if there was a big correlation between years of education and pathology in the brain that suggested a patient would later get Alzheimer's. So this specific line graph on the x-axis, 12 years of education means the individual completed high school, while 16 years means they completed a bachelor's degree, and then further means a graduate or a master's degree. So again, we looked at those four different cognitive groups that I mentioned on the very first figure. And here we can see that the data was kind of skewed, and there was a bunch of AD or Alzheimer's patients on 12 years of education and then also 20 years. So we didn't see a strong correlation or a strong trend. So this suggested that years of education wasn't a huge like kind of it didn't play a huge role or a correlation in showing Alzheimer's symptoms, but more it just provided that cognitive reserve before the patient actually got Alzheimer's compared to what they did. And then we also looked at education again with the second biomarker, which was that hippocampal volume. So this graph was actually kind of surprising because um, it did prove our thing with the ApoE4 status. As you can see on the graph, the teal line, meaning those patients did have the ApoE4 allele. They had a more decreased amount of hippocampal volume compared to the red line. But as you can see, it's actually a decreased overall. The more education a patient had, this line says that the lesser amount of hippocampal volume they had to if they had Alzheimer's. And we expected the opposite trend. Instead of a downward trend, we expected a positive trend. But that just shows that there's so much more other factors other than just education that were probably influencing the people with 20 years of education to get Alzheimer's in the first place. That's just another way we just tested different factors. So just to conclude, in our study, we explored the differences in various imaging markers. So we looked at white matter hyperintensity burden and hippocampal volume. And then we also looked at other aging-related pathology across individuals in those 4 different cognitive status groups, and then also education. So we found that non-carriers exhibited a broader spectrum of brain health ranging from very healthy to unhealthy, whereas carriers of the ApoE4 allele tend to cluster around a more uniform, less healthy state in all of the graphs that I just showed. And this condensed distribution highlights that the persisting influence of that A4 allele on brain health. And this just suggests that the ApoE4 allele does act as a very strong determinant for AD brain pathology or Alzheimer brain pathology. And it shows that it's a good prediction of whether an individual will get Alzheimer's. And these are the references that I used in my paper. And last but not least, I want to give a huge thank you to the 89 database. All of my graphs and figures were made using data from that database. So I'm really grateful that I was able to request that data and use it. Thank you so much to Talaria for being able to provide me with such an invaluable experience like this one. Being able to indulge in research and being able to conduct a whole research paper was definitely something new for me. So that was a really great experience. And then last but not least, thank you so much to my mentor, Krista. She was amazing through this whole process and she gave me so much help and feedback whenever I needed it. And I loved chatting with her each week and working on this project with her. And that is all for me.